take your chairs and settle down a little. Welcome, everyone. We're so excited to see you. Isn't it great to be all back together? I love it. Just to be with our family again. It's so exciting to be here. And I have to say that, I don't know, it gets, when you're in this field, you worry about the research and the science, like, because we're not getting face-to-face time with them, and you know, like, what's happening, what's going on, do they still care, are they still working on it? It's kind of like a mystery during this past 2.5 years, and you feel like you're in this black hole or whatever, but I can guarantee you, with tears coming in my eyes, that we are the luckiest organization in the universe to have these scientists and doctors taking care of us. They are awesome. We are very delighted to bring to you their work and what's been going on in the past 2.5 years. And I just have a couple of things that I'd like to say. One is, don't worry, we're not gonna have questions and answers during this session. So they're gonna just get through their talks. And in the afternoon, actually at 1 p.m. in this room, is Ask the Doctors for the parents, the pigmentos. Any pigmento around is invited to that session. So write your questions down. I did put on the corner of this table, there's a notepad and some pens on the corner of the dais here. If anybody wants to take a pen and a notepad just to be able to write your questions down. Otherwise, put it in your notes in your phone just to remind you of the question you want to ask. And then we're going to have a whole hour with them. We're going to have lots of time to ask all the questions that we want and really delve into their talks. At 2:10 is a PWA, Ask the Doctors. And, you know, while it's, you know, unusual to separate like that, it really was a request of the PWAs to be in their session alone. Years ago, I'm a historian here, it's in NOAA, been around for a while, but they wanted a chance to be able to just talk PWA to doctor without worrying about what us parents might be worried about, what they're thinking. Even though we love to hear their thoughts and what's going on, they wanted to have a private time with the doctors. So that's a PWA session only, and we will have bouncers at the door to keep everybody straightened out. The other thing I wanted to mention is this is a recorded session. This session will be available, and you'll be able to review it again. It is a recorded session, because sometimes I get overwhelmed, like, with what they're saying, and I get stuck on a word and I'm missing the next word. So this is going to be able to be seen again if you so choose. So it is my absolute pleasure. I mean, other organizations have scary doctors and scientists. We have the best. They're approachable, we can reach them. We get them in a moment's notice, they'll answer our questions. They're just really incredible. So I'm going to introduce our first speaker, and it's Dr. Murray Brilliant. Dr. Brilliant is semi-retired, probably less retired than he wants to admit. He's a PhD researcher geneticist with a long-standing friendship to NOAA and the HPS network too. And he's a worldwide albinism community. He currently works remotely with colleagues at the University of Wisconsin in Madison. And in the past, he has worked with Dr. King, Dr. Oding to uncover albinism genes. With Dr. Brooks and Dr. Summers, these are, you know, the best in albinism research. Dr. Brilliant, who is brilliant, by the way, developed potential gene therapies to improve vision for people with albinism. And with Dr. Carroll, began to understand vision in people with albinism as well. Over his 35-year career, he started when he was 12. He had discovered genes for OCA2, OCA4, and HPS1. Based on his research, clinical trials were launched, and the aim of improving vision in people with albinism has always been deep in his heart. I really, really have a long-standing friendship with Dr. Brilliant, and please say hello to Dr. Murray.
So I thought I'd start the session with basically sort of what we know about albinism and most importantly what we don't know. So I won't tell you, I won't give you data on what we don't know because we don't know it, but I will give you data on what we know. So what we know about albinism is we define it this way: congenital genetic abnormality of melanin synthesis. So that means, congenital means that it's present at birth, genetic means that it's inherited, and it's about melanin synthesis, melanin being the pigment in human beings that gives us our hair color, eye color, skin color. So this is a condition in which this amount of melanin made by these pigment cells, which are called melanocytes, is reduced or absent. And it always includes specific abnormalities of the eye. So we define albinism as having these specific abnormalities of the eye, and we recognize several different types which correspond to different genes. There's oculocutaneous albinism. Oculocutaneous means skin, hair, and eyes. Albinism, abbreviated OCA, I think most of you know that, but there are a number of different genes, so I just put a number there. But the main ones that we talked about are OCA 1, 2, 4 especially. There's also ocular albinism, which only gives us those abnormalities of the eye. And then there's albinism plus other features, notably the different genes and that underlie Hermansky-Pudlak syndrome. So what we don't know. Can we predict visual acuity in people with albinism? So far, we're trying to do that, but we don't have a very good algorithm or formula for that. And we want to also know the relationship between genetics and vision. We also need to understand the relationship between the anatomy of the eye and vision in people with albinism. And then to explore therapies, different drugs to enhance visual acuity. Can we do that? And if we can, what's the best age to utilize such therapies? So I'm going to give you just a very brief outline of what we call autosomal recessive. And there's an important advertisement here that I'm going to do a demonstration of genetic principles at 10:15 on Saturday. So if you want to know more about the genetics, please come to that. But in autosomal recessive conditions, which includes almost all forms of albinism, you have to have two parents who are carriers for the albinism trait in order to have a child affected with albinism. And typically that means that if both parents are carriers, there's a 1 in 4 chance for each child, each birth, for someone to have albinism. That means that most carriers who have children, they may not have any children with albinism, and so they don't even know that they're carriers for albinism. So the dogma that we have is that these recessive forms of albinism require 2 non- or low-function copies of the same gene. So that means that if you have OCA1, both parents have to have these variants in OCA1 in order to have a child with OCA1. They have to be carriers for that, for variants in the OCA1 gene in order to have a child with OCA1. And the same goes for OCA2 or 4, etc., HPS as well. So having 1 non- or low-functioning copy of an albinism gene means that you're a carrier, you don't have albinism. And again, the dogma is that you have to have these changes in the same albinism gene. And basically, that also means that not all of these changes result in a non-functioning gene. Sometimes it just lowers the amount of pigmentation. And so we do see kind of a range. As you can see, even looking around the room, different people with albinism some people have more pigment than others. So there's a range for various albinism genes, at least in terms of what we can, what we see in terms of their hair and eye color. So if pigmentation came in a cup, you know, you have different states here. You have albinism, where the cup is empty of melanin, or just a little bit of melanin. Carriers, where the cup is kind of half filled, and normal, where it's filled, right? And so we're beginning to understand that there are exceptional cases where somebody has a mutation in, say, OCA1, but a lot of other variants in other albinism type genes, and they may present as having albinism. So it's not always a single gene involved, although it mostly is. So the common features, the ocular features that define albinism are a reduction in iris and retinal pigment, iris being the front of the eye, retinal pigment in the back of the eye. Also, we see iris translucency. That means if we shine a light on the side of your eye, light can come out of where we see in the iris. Then, if you look in the back of the eye, there's in the back of the eye, there's a structure called the fovea, which I'll illustrate next. And that is not fully developed most of the time in people with albinism, although there are exceptional people with albinism that also have a fovea. There's reduced visual acuity, which all of you with albinism know. There's misrouting of the optic nerves at the chiasm in the brain that will disrupt stereo vision, depth perception. And then there are eye movements, nystagmus, and alternating strabismus. So if we look in the eye here, we see where the arrow is pointing to macula between the two pictures here. There's also a little cup there called the fovea, a little dent in the back of the eye, and that's where your best vision is. And this is underdeveloped in people with albinism. And that results in a degradation of signals to the brain from in your visual system, and a reduction in visual acuity. Okay. And then this is from 2008, NOAA. Just kind of illustrating how people with albinism might see. So that degradation you see 2020, 2200, 2400. How the image gets degraded when there are fewer photoreceptors in that fovea. Okay. And again, there's this misrouting of the optic nerve in albinism, resulting in differences in deficits in depth perception. And then, of course, nystagmus, which is that eye wobble. And just to illustrate a little bit about melanocytes, it's probably a little bit easier to show this picture of melanocytes in the skin. The bottom colored cell there is a melanocyte. Kind of looks like an octopus, and it has little fingers projecting into the skin. Inside that melanocyte, there's a factory that makes melanosomes, which are packets of melanin pigment, and that gets distributed into the skin. What's very interesting is that human beings, in how we are put together, and how the melanocytes kind of migrate in early development, it turns out that human beings are actually striped animals. We are striped. And this is illustrated by a rare patient seen here that is a genetic mosaic. Some of this person's cells have albinism. Some of the melanocytes have albinism, and some of them don't. This is what we call mosaicism. And this illustrates then the pattern that we see in human beings, that we're striped. But almost all of us, with the exception of these rare individuals, those stripes are the same. Okay. So for those of you who are very interested in a general overview of what we know about the genes and their functions in albinism, I refer you to this recent paper, Baker et al. In Progress in Retinal and Eye Research. It's just just been published. Anyhow, this kind of goes through this whole process of how pigmentation is produced and what the different genes, the functions of these different genes are doing. And to illustrate that a little bit differently, if we look at the proteins that are encoded by these genes that are associated with albinism, and we look at the melanosome, that's that pigment granule that is made by the melanocyte. What you can see here is sort of a working model of how these genes interact and kind of what they do. So I'm not going to hold you to understanding this and repeating this. It's a complicated process, but it goes to illustrate the detail that we now know about the genes that are associated with albinism, but also keep in mind that there's a whole lot that we want to know. And again, most of us, all of us here on the panel, are dedicated to trying to figure out what's going on with vision and albinism in order to come up with potential therapies. So I will stop here, and we can move on to our next speaker.
We're just going to introduce Dr. Adams to you, who is incredible, but I just want to say that wouldn't it be cool if we could actually drink pigment in a cup? Like, that would be cool. Like, eliminate the photophobia by having some Guinness or something like that. I'd say meet me at the bar tonight. Let's try it out. I don't know. Very cool. Thank you, Dr. Brilliant. Our next speaker, I also want to mention, there are people standing in the back, and if somebody, in order to facilitate this, if somebody's sitting in a seat that is next to an empty seat, could you stand up so that people in the back can come and sit in some chairs? There we go. Just have people fill in. Good job. Okay, that's great. I think we're good. Thank you very, very much. That was a great exercise in albinism learning management. Our next speaker is Dr. Adams. We're really very grateful that he's here. Dr. Adams is a pediatrician, medical kineticist, working at the National Human Genome Research Institute in the lab of Dr. William Baum. He received his MD and PhD from the University of Washington in Seattle. His interest in albinism was inspired by Dr. Ball's work on several medical conditions that include other diseases of hypopigmentation, one of them being Hermansky-Pudlak syndrome. He started an ocular cutaneous albinism natural history study, yay, at the National Institutes of Health in 2008. His current work focuses on albinism genetics and the use of cell biologic techniques to study human pigmentation and develop new therapies for OCA. Dr. Adams' other clinical research work focuses on the application of large-scale genomic analysis techniques in the diagnosis of rare and undiagnosed diseases. We're very excited to have Dr. Adams with us. Everybody welcome.
Great. Thank you, Donna, is always very nice to us in our in introductions, but I think everyone up here agrees that it's really our privilege to be able to speak with you and to work with you around albinism and partner in trying to find some solutions and ways forward, both to understand albinism and hopefully to improve the lives of people with albinism, which is our ultimate goal. I guess the other thing I wanted to mention is that we are frequent flyers at this meeting, but we're not the only people that are studying albinism, so I hope that that doesn't come across. There's really some wonderful research going on around the world. John Zippin in New York, and my colleagues in the NIH intramural program, and researchers in other parts of the country. So you're getting kind of a sample of what's going on, but just so you know, it's not everything, which is great. We'd always love more resources, but there are some exciting things happening. So I'm a pediatrician and geneticist. The NIH funds research across the country. About 10 or 15% of the money that you pay as taxpayers to the NIH funds a large facility in Bethesda, Maryland, and the rest of the money goes out to universities where all sorts of research is undertaken. So I work in that intramural program in Bethesda. Some past work, I had a natural history study that was open from 2009 to 2019. I'm extremely grateful for people in this community who participated in that study, and we learned a great deal. And those families really selflessly told us their stories, gave us skin cells. You know, there's some folks in the audience with small scars on their arm because they were willing to let us take a skin biopsy. We're still using those to try to understand albinism. And they told us their stories. And although that study ended in 2019, we're interested in thinking about what the next study should show us, especially something that will help us to move forward towards treatment studies. We did one treatment study, which was a medication called nitisinone that just included 5 individuals, and so it was very preliminary, not enough to really start thinking about using that medication. And we learned quite a bit from those 5 brave people. There was some increase in pigmentation in skin and hair, but the signals were very small. So we're thinking about where to go next with that study, whether people who were younger, these were all adults, or changes in the study design. Of course, the pandemic happened and we stopped traveling around, and I'm sure just like you did, kind of holed up in our houses. We're all kind of rusty this morning. You'll see that we're, getting stuff loaded is going to be a bit of some work this morning. So we're right there with you in terms of, sort of in recovery from this strange few years that we've been through. So at the NIH, we decided to, instead of working hard and developing another clinical protocol or study that we would invite people to participate in if they were interested in, we really focused on samples that we had in hand and tried to progress that way. And in particular, we combined samples that we had received from participants in the natural history study with a very large collection of DNA specimens that were donated by people who had worked with Dr. Oding and Dr. King back in the day. So older folks with albinism may remember Dr. King. And Dr. Oding continues to be a part of our research group. So he calls in to our meetings every week as we think about the genetics of albinism. So not every change in our DNA causes health problems. In fact, most of them don't. So I spent a lot of time looking at the sequence of our genomes for health and research reasons. And any 2 of us in this room would have 2 to 4 million differences between our genomes. And of course, most of those aren't causing health problems. Some of them contribute to the fact that we look different, we have different heights, for instance, different ear shapes, but many of them we don't understand the differences. So the question is how can you tell if a change out of those millions of changes is actually important for your health. And that may be your risk of heart disease later, or it may be related to albinism. So if a person with albinism decides to have genetic testing, the testing may fail to provide an answer. And I'll have to say that this is probably the most common question that I get from other doctors who call me about albinism is, a person clearly has albinism, they've come to me and they would like to have testing done, and we do the testing and we either come up with nothing or we come up with not enough changes in the DNA to explain the albinism. And you might say, why is this, why is it important? And different people have different reasons, which I don't second guess for wanting genetic testing. But for research, it's super important. So I mentioned that there were 5 people in our study that we gave this drug patissinone to and looked for pigmentation changes. And that entire study was sort of predicated or based on the idea that those individuals would have type 1 albinism. And that's just because of how that particular drug works. Turns out, after the fact, after the study was completely done, one of those people had albinism that was caused by a completely different gene. And the reason why we didn't realize that is because the science hadn't gotten to the point where we were really adequately testing all of the albinism genes. So while that was interesting, it certainly, in terms of the study, makes you realize that you need to build your projects on a firm foundation, and really understanding albinism and how it goes through families is one of those. So why do these tests fail? In some cases, it may be that we're not testing the right genes. So since we started coming and presenting at NOAA, there have been some new genes that have been reported as causing albinism. They're pretty rare. Our tests may fail to detect some types of changes in albinism, and I'll talk about that a little bit more. And then, in some cases, we might see a change, but we don't recognize that it causes albinism. So again, there's this sorting process. We're looking at a million things and trying to decide which are the relevant ones. And if we don't have that genetic information as medical providers, it makes it difficult to provide adequate answers to families when they come and ask this question. So, does my child have albinism, especially if it's a mild case? Is this a type of albinism with other health concerns? And I think Donna has done a wonderful job of advocating for HPS, and it's very important to understand what you need to look out for in your life if you have albinism. Is it just going to be vision and the eye issues that you'll work with, or are there other concerns as well? What will happen with future children in our family is a question we get asked, and if my partner and I both have albinism, or will our children have albinism? And all of those questions are more difficult to answer if the tests fail. So I just wanted to show an example of what I'm talking about. So here is a sentence example, and the sentence is, the color of the sky was a surprise. Now I've changed the sentence, so I've spelled color, C-O-L-O-U-R, and I've spelled surprise, S-U-R-P-R-I-Z-E. So these are different spellings, but the meaning's clear. I don't think any of you would look at this sentence and say, I don't understand what it means. There are variants, and in other parts of the world, they're more common, especially the spelling of color. And so these are variations that we're kind of used to seeing in our language, but again, doesn't change the meaning. So here I've changed the sentence to, the color of the ski was a surprise. So I've only changed one letter, sky to ski, S-K-Y to S-K-I. But now the meaning of the sentence has changed entirely, right? I mean, it means something different. And this is the same thing that we struggle with DNA, although we don't have the knowledge of DNA sequence in many cases that we have of language to be able to make an interpretation here. So as you can imagine, there's sort of a spectrum of changes, right? There's some changes that we can say that isn't doing anything, and there are some changes that seem to be very clearly changing how the gene functions. So, for instance, if a variant is very, very common and lots of people have it, it's probably not causing a very rare medical condition. However, there are other variants where it's uncommon, and so it could be causing a medical condition when we look at other types of evidence. So our work, and there's a student now, a staff person with our group, Linnea Lund, who is joining me at the meeting today, and she's done a ton of this and just gave a presentation to our lab group about it recently. But we have a set of rules that we use to try to go through and understand any specific change in the spelling of one of our genes, and that puts us into one of several categories: yes, maybe, or no for any given difference. And so much of the current work that we've done for the last couple of years, getting back to where I started, is trying to refine the set of rules that we use to classify different changes in albinism so that we can give better information back to people if they get testing, and we're working with them to interpret the results. So we're adding to the list of variants. We're looking for exceptions to the rules that are really specific to albinism. So there are rules that we use for genes that cause all sorts of different things, but how can we customize those to the albinism gene so that we can make better interpretations, and then also proposing improvements to testing approaches. So here's kind of an interesting example. I have a couple of figures, and I know that these don't all scan well, depending on folks' vision. I'll do my best to describe them. So there's 4 bars here. The top bar has two DNA changes in it. One is, that should be S192Y on the left, on the right. S192S is the normal configuration for that position. R402R is the normal configuration on the right. And then if you look at the second line, you can see that one of those has a change. So the S has changed to a Y. So this is simply a change in DNA that's changed one of the components of the protein that's made. And in this case, this is tyrosinase, which is associated with type 1 albinism. On the bottom line, there's a different change on the right-hand side, which is purple in this case, and that's R402Q. So those two changes, the S192Y on the left and the R402Q, are both very, very common. Lots of people without albinism have those changes. And as a matter of fact, you can have 2 of those, one on the gene you got from your mother and one on the gene you got from your father, and still not have albinism. So it puzzled us that these show up a lot in cases where we test a person with albinism and we can't find an answer. We find one change that looks like that's really important, and then we look for the second change because you need 2. And we can't find that second change. And instead we find these. But how can this possibly be important? Because again, they're so common. It turns out that if you have both of those changes on the same piece of DNA, so this is either you got them both from your mother or both from your father, that may mark a gene that actually doesn't work as well as it's supposed to and might contribute to an albinism diagnosis. So this is, we call this the YQ line, because one position changes to a Y and the other one changes to a Q. And we have been looking at large groups of individuals with and without albinism to understand how we can provide advice to families who have this change. Right now, these are not being tested for in routine testing for albinism, so this also feeds into that goal that I mentioned of trying to improve testing. So it might be that you get to do the standard testing, doesn't come up with anything, and then you go to look for this afterwards to see if it helps explain things. Another thing that does not, isn't detected by current testing are complex patterns, and this has a lot of busy detail, which is not all that important. The main thing are the 2 black lines on the bottom. So, as Dr. Brilliant and I had mentioned before, you get a gene from each parent, right? So the normal number of copies of the albinism gene that we have are 2 for most types of albinism. But in some cases, there's a piece that's missing, or there's an extra piece. And in the bottom 2 black bars, you can see that there's some notches, and this is a very complex change in type 2 albinism that results from some pieces that are either duplicated or missing in the gene. And again, this is a case where the typical testing we do wouldn't pick this up, and so you would need to do some sort of expanded or additional work to determine that a person's albinism was caused by this change. And then finally, there is a deletion that's very well known in the albinism community that probably arose in Africa. And you have to do some careful testing to find this, and it's certainly worth testing for if a person has albinism and they have some African ancestry. That's on the left-hand side. On the right-hand side. On the left-hand side is another one of these that we found that showed up in a different part of Africa. And so this points to the fact that not all of these results are going to be the same for every person. So thinking about what a family's personal history is, what their ancestry is going to help us to be able to design testing that is more likely to yield results. So for future work, we're looking at changes in multiple genes. We would like to complete a formal submission of our findings to a place where other doctors and testing laboratories will use them. And then we would like to think about future clinical studies as well, especially studies that focus on how, if we gave a medication and it helped things, how would we know that? What measurement would we make to show that there had been an improvement? And that's actually been a fairly challenging task. So I just wanted to point out Stacy Loftus, who's done a lot of this work. I work very closely with Linnea, who's a student heading off to genetic counseling school, and she's done a lot of work on this project. And William Oding, who has really done a wonderful service to the community, I think, by transitioning all of the work that was done by Dr. King in terms of the DNA that was collected there to this current work so that it wouldn't get lost and we could continue to try to use it to the benefit of the community. Great, thank you.
Thank you so much, Dr. Thank you very much, Dr. Adams. I didn't break anything, just want to let you know. I just really appreciate, I really love his analogy about sentence change, like sky and ski and variation. That was very cool. I think that really, to be able to bring it to us so that we really get to understand it better is a talent, and I thought that was really good. I also want to mention that, he certainly kindly mentions that it's everybody's personal ideas of whether or not they want to gene test themselves or their family. It's a personal decision. But as he said, in science research, it's very helpful. So I just want to mention there's a lot of opportunities this weekend to kind of discuss that with people. Certainly, as a nurse and a mom, I really recommend everybody ruling out Hermansky-Pudlak syndrome, just as a point of reference. But also, there's going to be iGene is here. They're going to talk about how to get gene testing. Rare Genome Project is going to be mentioned at my table. So there's a lot of ways to be able to get gene tested, as well as at the Ask the Doctors. You can discuss that with these doctors. So make sure you use the weekend as an opportunity to decide what you want to do and learn how to do it. Our next speaker is Dr. Stroll, and we're very excited to have her with us. Dr. Gail Summers was with us for years, and she was wonderful and very kind to us all. But she blessed us with her protege. She blessed us with the sustainability of her work in Dr. Stroll. So she is here with us. She's a pediatric ophthalmologist from the University of Minnesota. She's the former partner and preeminent expert of the preeminent expert, Dr. Gail Summers. And Dr. Summers inspired and mentored her to continue to advance care and research for patients with albinism. Dr. Stroll inherited the great privilege she wrote this, so she named it. The great privilege of the directorship of the International Albinism Center at the University of Minnesota. Her work has led to international collaborations, including contributing to the creation of the first international albinism database with major albinism centers across the world. She also is proud to be a co-investigator with Dr. Carroll, that you'll hear from next, which brings valuable insight into the anatomic basis for the reduced vision in patients with albinism. And her greatest joy has been in patient care and has motivated her to search for answers to inform and help us. and she's truly very, very kind and sweet. So.
Hi, good morning. I'm going to keep my mask on, so if you're having difficulty understanding or hearing me, I'm not usually accused of being quiet, but please just raise a thumbs up so I know to be a little bit louder and clearer. It's really nice to be here and be a small part of NOAHCon and to see some familiar faces, as well as lots of new ones. I'm going to dive right into the update that I'm giving, which is focusing on one part of the eye that changes in albinism. And you know, Dr. Brilliant really gave a really wonderful introduction to so many different anatomic changes in patients with albinism. So this is that one small slice. We're going to focus on foveal hypoplasia and a little bit, just a slice of the work that's being done in that. In order to understand what is going on with research though, we got to start with what is foveal hypoplasia. So I thought I'd start with showing you what a normal fovea looks like. So when you come in to see me or your ophthalmologist or seeing your child, what we're doing is we're dilating the eyes, everybody's least favorite part of the exam. I know, I have to dilate every year too. I always tell my little kids, I get it. And when we look back there, what we want to see is the retina, which you see here. The optic nerve, which is to the side here, that is the cable that connects the eye to the brain. From it branches blood vessels that supply the retina with nutrients and oxygen. And in the center of those two blood vessel arcades, you actually see something called the macula. And right in the center of the macula is the normal fovea pictured here. So this is a patient not with albinism, but with a healthy, fully formed retina and fovea. And you can see that right in the center there is a tiny little reflection of light. And that is representative of that dip that Dr. Brilliant had mentioned. And so even though this is a two-dimensional photo, we know that there's a three-dimensional dip here. We also see that there's a surrounding light reflex. That's a little bit farther out from the fovea. But what's important to know about is that very central part where that light reflex is what's serving our most central, our most fine and detailed vision. So when we have patients with albinism who come in, they might look like this. And for years and years, thanks to the work of Dr. Summers and others, the way we categorized and understood severity of foveal hypoplasia was by grading them based off of what we saw on the dilated exam. So here you have the fully formed fovea on one side, and to the other side you can immediately notice some differences. So this is a patient who has the most mild form of the foveal hypoplasia. So again, that means just underdevelopment of that central 20/20 spot of the retina. And you can see that there is some good pigment here. You're not seeing any blood vessels underneath the retina, but what you're not seeing as well is that light reflection. Neither that area that's that big circle nor the tiny one in the center. So this is a patient who has some pigment development, so it's more mild, but we're not able to see that that fovea developed fully. If we go to the more severe forms, we get to grade 2, and that's where you can start seeing some underlying blood vessel network underneath the retina. So you're going from a very opaque retina to a more translucent one. So you'll start noticing that you're seeing some differentiation in coloration. It's most obvious that we're having translucency or really transparency in the more severe grade of foveal hypoplasia. So here you're seeing the retinal vessels coming from the nerve, but you're also seeing a network of supply blood vessels that live underneath the retina. So truly there's no pigment in the retina and there's no foveal development here. Now this worked really well for us for some time to just give us a sense of severity, but it didn't really match well always to what we were seeing for visual function for our patients. So it just gave us a generalized sense. But about a decade or so ago, we actually got some new tools. The fun thing about ophthalmology, in addition to all the patient care, is also the research and technology. And we got this amazing thing called OCT, optical coherence tomography. What it is it uses light to actually take a scan of the retina. And instead of looking at just the top of the layer cake, which you can see on exam here, that's all we can see is that top layer of the retina, it actually takes a slice of the layer cake. So you can imagine here, we have just one line that that OCT machine might take, and then what we would get is the image to the side here. And you can see all those layers. And so we actually can see how developed or underdeveloped that fovea or that retina is. And this gave us a lot of information because we can compare it to what we think of as a fully formed fovea. And so again, you can see what Dr. Brilliant talked about is that fully formed fovea has that nice dip. you can immediately notice that that dip is not present in this patient who has foveal hypoplasia. If you look closer, you'll also see that other layers are not correctly pruned or not correctly widened, and so all the layers can be different. So why did we focus on foveal hypoplasia for research when it comes to albinism? Why is this research so important and something that I'm really excited about, and that you hear a lot about in all of our talks probably? Well, the reason why is right now it's the most strongly associated with visual function. So structural changes throughout the eye and the brain exists in different parts, but we know that the level of foveal hypoplasia will be the best predictor for visual acuity. And so there's been amazing work on this in the past. I commend my colleagues who have really built such an incredible foundation. So Dr. Carroll and Dr. Brilliant, who are here, along with Dr. Summers and others, actually showed us how much variety in foveal hypoplasia there is. And I like this because it's just a zoomed in part of one of the figures in their article, in which you can see the variation in foveal hypoplasia. So all these pictures are an OCT slice from a patient with albinism. And you can see that right here, we've got one patient who has a little bit of a foveal dip, but right next door we have a patient who has no foveal dip. Here we have one who has this thickening of the bottom layer of the retina, but here we have no thickening. It looks the same throughout. So there's a ton of variety. And so we started thinking, how do we understand a little bit more about the variation that we see? So as you heard, we are not the only researchers or those interested in albinism, which we are so thankful for. There's brilliant people around the world. And out of the United Kingdom, the Leicester system came out of the University of Leicester group. And so what they did is they actually used the OCT to create a new grading system for foveal hypoplasia. And so now, instead of just looking at the top of the layer cake, we could look at the layers and be much more detailed in how we determine the severity. So we actually created, well, they created grades 1 through 4, as well as atypical. And what they did is they actually showed that it can help with predicting visual acuity. So what they found is that if you had the more mild form, grade 1, what they consider mild severity of foveal hypoplasia, the median, the middle visual acuity for patients that they studied was really good, 20/32. So remember, we consider normal vision to be 20/20. Well, as you go up in severity, they saw a corresponding level of reduced vision. So grade 2, the median was 20/55. Grade 3, the median was 20/80. And grade 4 was 21/25. Now, those of you sitting in the room might be thinking of your family member and how they might fit into that category where they kind of land. And it's possible that that's a major part because of their level of foveal hypoplasia. So after this came out, there was a lot of interest, but we also found that there was a lot of overlap. And so we actually went on to do some more collaborations. So we were lucky enough at the University of Minnesota to be one of the collaborators with this international project that just came out this year. And what we did is we actually had 12 different centers in 9 different countries around the world that actually looked at not just albinism, but all different types of things that cause foveal hypoplasia. So the part I'm going to focus on is just the part that affects patients with albinism. And again, when we look at this picture here, I really love this figure because it kind of drives home the development and the interruptions that are possible in foveal hypoplasia. So in A, you're seeing all the layers of the retina before they become the fovea. And what we normally want to see is we want to see that there's an area where it's pruned in the center, and that becomes more and more thin in that top layers, while the bottom layers that are these cone cells, the cells that actually serve our finest vision, that take light and turn it into electrical activity to be interpreted by the brain into vision. So those numbers increase, while the layers on top of them are pushed to the side. Now, why is that important? Well, the light that goes into all of our eyes would normally, if you didn't have this little pit development, have to go through all these layers in order to get to the back part of the cones to actually be translated into electrical activity. So to serve our finest vision, we actually lose those extra layers so the light is not interrupted as it's getting back there. So you can imagine if there is those layers in front of it, if the fovea did not fully develop, if you have foveal hypoplasia, vision might be reduced simply because of that, or it can be reduced because the cones here are not as dense. So the cones can be, sorry, I'm just going to repeat. So we can have reduced vision either because we have, or both, more layers of the retina to get through for the light to actually penetrate to those cone cells, or because we didn't fully develop a density of cone cells right here. And they showed this to be the case when they looked at patients and they tried to grade them by their OCTs. And they found that there was really a significant change across different types of albinism. Now we're just going to focus on the grading of grades 1 through 4, since patients with albinism do not have atypical foveal hypoplasia. So this box plot here, the big thing to know is that starting from grade 1 to grade 4, the higher up you get in this scale, the worse visual acuity was. And what I like to know and notice here is that there's overlap between them. So grade 4 patients had worse visual acuity in general from patients who had grade 3 foveal hypoplasia. But there's patients in grade 3 who have worse visual acuity than some patients with grade 4. So where there's not a really clear delineation, we're not able to say you're definitely going to be this level, right? And so this is a good categorical system, but it's not perfect. So what did we learn from this study and from the work that's being done here? Well, we learned how genes relate to phenotype. So genes, remember, is the DNA, the mutations that we see. Phenotype is how you function every day with your visual function. And what we really learned is that there is a wide spectrum of foveal hypoplasia and albinism. And so we saw that there is likely a variable time frame. So different genes are causing disruptions at different time points in the development of the eye. And that is likely what's causing all this variation that we're seeing. The big take-home messages that I find really helpful in talking with families and things that get to the nitty-gritty of visual acuity and prognosis is that HPS and ocular albinism was actually associated with worse grades of foveal hypoplasia, meaning worse visual acuity. Whereas those with oculocutaneous albinism had a wide spectrum. And so that's really important to know. It's something that you guys might experience yourselves, because you might have children, siblings who have oculocutaneous albinism, and you're noticing that their visual acuity outcomes are different. And that's something we still don't understand. So I'm very excited about this work. You know, an ocular coherence tomography picture, it is not too hard to get. So we could probably get it in patients as early as about 3. I have had a precocious 2-year-old who can do it. But the issue is that in order to get that information, you have to have not as much nystagmus, that eye wobble, in order for the picture to not be blurred. And so this study, as well as clinical application right now, is limited by the fact that we don't have a good way to take those pictures unless, of course, we sedate you. And that is a big issue, something that we still have to solve. The other thing that I would really like to do is get more down into the nitty-gritty, and something I think we need to look ahead to the variation in oculocutaneous albinism and start really understanding the different types of oculocutaneous albinism and visual outcome related to these structural changes. With that, I'm going to leave you guys in the great hands of Dr. Carroll. Thank you guys so much for your time.
Thank you, Dr. Stoll. Dr. Carroll actually wanted me to almost eliminate his bio because he wants to get to the meat of the potatoes here. But we want to really tell you that he is really wonderful. He's a professor of ophthalmology and biophysics, biomedical engineering, and cell biology, neurobiology, and anatomy at the Medical College of Wisconsin. Oh, yeah, Debbie, can you come up? Thank you. Call for Debbie again. Debbie is our savior over here. Thank you, Debbie. Dr. Carroll directs an imaging program which utilizes a variety of retinal imaging tools to study the structure and function of living human retina. His group has worked to develop image analysis approaches to increase the scientific and clinical utility of these imaging devices, and has pioneered the dissemination of this technology to the research groups absolutely around the world. So he is, an amazing image guy. But I really want to finish his bio quickly by saying that NOAA really is indebted to you with the work that you do to help us with the new NOAA research program. You have been very kind, and we're very, very grateful.
Well, thank you very much. I've been working on this for 13 years. This is my first NOAA convention, so And it's an honor to work with NOAA and the individuals seated at the table here. And I want to just end this session with some updates on research that we've been doing. We talked about science. There are a lot of people working in albinism research. It's a very collaborative group. This is my team in Milwaukee. I think I saw a Brewer jersey in the audience somewhere. Go Brewers, there it is. It's one of the least competitive domains in ophthalmology. Some areas of research, you see a lot of competition between researchers. I will tell you from my perspective, I called Dr. Summers when I was first starting my lab and said I wanted to learn more about albinism, and she spent hours upon hours upon hours, and that started our collaboration. They're a very giving community, and I couldn't ask for better colleagues, and some of them are listed here who've helped us over the years. But this figure that was just shown keeps me up at night. And it makes me come in the morning, because there's a lot of things here that I don't understand. And it was mentioned that someone with a grade 3 phobia could have slightly better or worse vision than someone with a grade 4 phobia. But moreover, someone with the best phobia, the grade 1, can have worse vision than someone with the worst phobia. That kind of thing just bothers me. It means that we don't know everything. And so that's sort of where I come from when I think about exploring what is it in our retinas, everybody's retina, that dictates what we see. And we saw this cross-section of the eye, but if we actually look at what is the retina, it's this picture on the right. It's a little piece of saran wrap of thin cling film, about 300 microns thick. And that's the tissue that is looked at in the clinic, that we study in the research setting. And it's really tough to study, because inside of that, it looks transparent. But there's about 100 million photoreceptor cells. Over here on the right, I've shown a little fancy electron microscopy image of photoreceptors. There's long, slender ones, which are the rods that help you see at nighttime. And there's a couple of short, skinny ones. Just very down here, just one of them. They're only about 5% of your photoreceptors, are those little short cones. The rest are rods. But yet those cones are what help you see color, which help you read high acuity information in what you're looking at. So it's very confusing that they're actually the minority of all of your photoreceptors, are arguably the most important ones. But they're not arranged uniformly across your retina. The fovea that you've heard a lot about is actually where all the cones are packed in really tightly. So the most important cells are in the most important region of your retina, and that makes some sense. The rods tend to be out in the periphery. They help with your side vision. And so what we've understood from sort of a computer science perspective is, how do those cones limit what you see? Many people might recognize the eye chart on the left. What I've got on the right is a picture of, let's just call those cones. Each one of those circles is a cone. And I've put a letter E from an eye chart onto the cone mosaic. I call it a cone mosaic. Now, for a given retina to reliably say that that's an E, there have to be a certain number of cones where the black part of the E falls on, and there has to be at least one row of cones that occupies the gap between the bars of the E. That's a sampling theorem that we know about that dictates the best resolution you could hope for if your cones were that size, spaced apart by that amount. That's the smallest E you could ever resolve. That's a theoretical limit of vision for that cone mosaic. Well, we actually have technology that lets us image the cone mosaic in people with albinism and people without albinism. I'm showing a picture over here. We use adaptive optics technology. This is from astronomy, but it's been moved over to ophthalmology. And it corrects for some of the imperfections in the eye and gives us a high-resolution picture of the back of the eye in the retina. Instead of looking at the layers, we look at one specific layer, that is the photoreceptor layer. These pictures over on the right, The ones on the left are actually from a microscope. So someone donated their eye, we took out the tissue, and we actually imaged those cells under a microscope. The ones on the right are actually a living patient, completely non-invasively. We can image the exact same cells. We can count them, we can measure their size, and we can measure how far apart they are. And here's an image of a patient's fovea, a patient with albinism, their very central fovea, where we can count every single photoreceptor, every single cone photoreceptor in their fovea. Surprisingly, and you don't need to know too much about this graph, except for that lots of the data seem to overlap. Well, this is combined data from people with normal vision and people with albinism. And they overlap. This shows that in many people with albinism, their cones are completely normal. That doesn't make any sense. So we have to ask ourselves, what else affects acuity? And the reason the cones are so important is they get a private line, almost a one-to-one cable, as you heard Dr. Struhl mention, to the brain. One cone to one ganglion cell neuron, all the way to your visual cortex. That's the private line. That's the normal situation. Rods don't get that. It's about 1,000 rods to one ganglion cell neuron. They're not that special. What we can also do, instead of using our adaptive optics camera to take a picture of the photoreceptors, we can actually present one of those eye chart E's directly onto the cones in a patient with albinism. And we can ask them which way the E is pointing. And then we can make the E a little bit smaller and ask them which way it's pointing, and smaller and smaller and smaller and smaller, until they just operate at a chance performance. Okay? And that's called finding someone's visual acuity threshold. And everybody has one. There's a point at which the E is no longer distinguishable. In a normal situations, that's governed by the size and spacing of those cone photoreceptors. And so we can derive from the image a theoretical prediction of how well someone should see, and then we can actually measure what they do see. And comparing those two things teaches us something very important about albinism. This is just what some of our raw data looks like, and it's not really all that important. But what I'm showing here is there's a dashed line. That'd be what we call a one-to-one relationship. That would be if the prediction matched our observation. And that's what we would expect to see. And there's some blue dots. The filled blue dots are for people with normal visual acuity, and they're almost right on that one-to-one line, which is what we thought we'd see. The open blue circles are from a couple of individuals who were born prematurely who have foveal hypoplasia. And the orange dots are from a couple of people with albinism. They fall below the one-to-one line. And what that shows us is that their performance is worse than what the cones say they should have been able to accomplish. Again, telling us that it's not the cones that are the problem. It must be something else that's affecting their vision. And our idea is that it's this one-to-one connectivity, that private line, direct phone, the red phone that the president has to call whoever he wants. Those cones are no longer connected in the right way to the brain. And so I've just drawn sort of a cartoon here that might emulate that, where you might have one neuron, one of those ganglion cells, is actually getting information from two cones. So that's how the information gets blurred right within your retina. And it's really important to know, is the problem with the cones or how the cones talk to the brain? Because it points us to completely different therapeutic approaches, right? If the cones are normal, we don't need to grow more cones. We don't need to pack more cones in the fovea. We need to focus on understanding how to teach them to talk differently to the other cells in the retina. That surprised me. One last thing. I talked a lot about cones, but remember, they're the minority. They only really do visual acuity, and they're only 5% of your photoreceptors in the first place. So what about the rods? It might be that rods could have a role in other things like nystagmus, light sensitivity, refractive error, etc. And no one's ever really looked at rods in albinism, although people have proposed that patients with albinism might have about 30% fewer rods than normal. But that was based completely on animal studies, and no one had ever looked at patients. We did that, And our data so far suggests that people with albinism have completely normal rod photoreceptors. We've only imaged 9 patients. We're looking for more, but so far, that's what research is. This is our research in progress, and I certainly look forward to telling you more about that in the future. I'm very fortunate. Dr. Adams mentioned that your tax dollars support research. Well, we were just awarded a large grant from the National Institutes of Health in collaboration with Dr. Struhl. You're very welcome to email us if you're interested in participating. Fortunately, there's travel funds involved to cover people to come and get imaged. It's super cool. We can see what's going on with your rods and cones. And lastly, if it's okay, I'll just share, thanks to the support of NOAA, we do have a survey. We're very interested in learning about people's experiences with access to genetic resources. This will just take you to a survey link. There's also a link on the NOAA website. You can go directly there in the research tab. This is something that we think is very important, as Dr. Adams' work was touching on, understanding how people in different parts of the world and different parts of the country actually can access genetic information is certainly something that's going to be important in the years to come. But with that, I'd like to thank you for your attention and look forward to this afternoon's Q&A sessions.
One more round of applause to all these researchers. Thank you very much for attending, and make sure you can come to the Ask the Doctors. Make sure you get the right one. Pigmentos in one, PWAs in the other. And join us this afternoon with all of your questions. Thank you very much for attending.